The proteasome is a large multi-subunit complex that mediates the degradation of a wide variety of proteins in eukaryotic cells. The proteasome consists of a 20S core particle and two 19S regulatory particles that cap either end of the core particle and regulate entry of proteins to the proteasome core. The core particle consists of two alpha rings and two beta rings, each containing 7 structurally related subunits to form an alpha-beta-beta-alpha cylindrical complex. Inside the internal chamber of the proteasome are 6 protease active sites located near the amino termini of the beta-1, beta-2, and beta-5 subunits. When a polypeptide is delivered to the core particle, it is cleaved into small oligopeptides that are released into the cytosol and eventually further degraded into individual amino acids. Protein degradation by the proteasome is a carefully regulated process. Target proteins must first be tagged with a protein ubiquitin on lysine residues. Ubiquitin contains multiple lysine residues that can serve as covalent linkage sites to other ubiquitin molecules. A chain of ubiquitins linked between glycine 76 of one monomer and lysine 48 of an adjacent monomer typically mark a protein destined for the proteasome. 3 classes of proteins are involved in ubiquitinating target proteins. These proteins are known as E1, E2, and E3 enzymes. First, ubiquitin is covalently attached to the E1 active site. Next, E1 associates with E2 and transfers the ubiquitin to the E2 active site. The E2 ubiquitin complex then binds to an E3 protein, also called an E3 ubiquitin ligase. This complex binds to specific target proteins, and the ubiquitin is transferred to the target protein. The target protein must undergo this reaction at least 3 times before it can be recognized by the proteasome. The 19S subunit of the proteasome recognizes and binds to the polyubiquitin of the target protein. Through mechanisms that are still being resolved, the proteasome uses ATP to release ubiquitin, unfold the target protein, and direct the polypeptide to the internal chamber for degradation. This selective mechanism for protein degradation is crucial for the regulation of numerous signaling pathways, as well as clearing misfolded and truncated proteins from the cytosol.